Good morning, everyone. So in the last two lectures, we covered how B cells get activated and then what happens to them. Now we're going to switch to the T cell side and hopefully draw some comparisons about what's the same for T cells and what's different. So we start with the bonus question from last time, which was, there are patients who have a deficiency in CD40 or CD40 ligand. It gives the same phenotype. And remember, that's the key signal for a T cell telling a B cell, yes, you should go form a germinal center. Well, the germinal center is where all of those things, hypermutation, affinity selection, and isotype switching occur. And so without that, the patient doesn't form germinal centers. Instead, they form the primary foci, which is just mainly IgM, and it's relatively low affinity. And they also don't get long-lived plasmablasts long-lived plasma cells or memory B cells. And so patients that have a deficiency in the CD40 pathway have what's called hyper-IgM syndrome, where they make IgM, during infection, but they don't make the response better by affinity maturation and somatic hypermutation. They also don't tailor the antibody response, so you only get IgM. And so these patients are susceptible to recurrent infections, and particularly with bacterial infections that can spread, resulting in sepsis. So that CD40 ligand signal from the T cells says form the germinal center. Without that, you don't get somatic hypermutation, you don't get affinity maturation, you don't get isotype switching, you don't get long-lived plasma cells or memory B cells. Okay, so that's why patients with this have recurrent bacterial infections. Okay, so let's start with T cell immunity. And I want to start with the questions, right? It's important to phrase this in terms of, how does the antigen get to the lymph node for presentation to T cells? How do the T cells get to the lymph nodes to look for antigen presenting cells? And then what signals do those antigen presenting cells provide to the T cells for them to become activated and go and do their job? And in the subsequent lectures, we'll start dividing this up into the different ways that CD4 cells and CD8 cells respond. But for now, we're just going to keep it general, talking about T cells as if they're one kind of T cell. Okay, so you remember that T cell progenitors, or precursors, come from the bone marrow and they go to the thymus. And this is where they rearrange their T cell receptor. They undergo negative selection for positive selection so that they can, in fact, recognize self-MHC, and then negative selection so they delete autoreactive cells. And then those naive T cells, when they leave the thymus, will then be mature CD4 or CD8 positive T cells. And those will be circulating through the lymph nodes and the spleen, which is essentially the lymph node for the blood, looking for antigen. And so we have actually measured, how many cells in a human or a mouse is specific for any particular epitope. In this case, an epitope on MHC. And it's about 1 in a million to 1 in 10 million. Mark Jenkins at the University of Minnesota has done some more work on this. Basically, there are very few cells that are specific for any given epitope. And so this is going back to the clonal selection theory, that there's 1 in a million is specific for any epitope, and that's the one that's going to respond. Now, these naive T cells that come from the thymus, they're mature, they're just circulating from the blood to the lymph nodes and back again to look for antigen. So they spend most of their time in a lymph node, or in this case, in the spleen, if it's looking for a systemic infection. And so it takes a T cell about 24 hours to go through a lymph node. T cell activation, though, has to, just like initial B cell activation, initial T cell activation has to occur in those secondary lymphoid organs, either spleen or lymph nodes, when the T cell encounters antigen on a dendritic cell. And so that's what we're going to focus on in this class. However, there's a difference here, right? B cells get activated on follicular dendritic cells. that are holding antigen on their surface, either by, excuse me, by Fc receptors or by complement receptors. For T cells, the activation is different on different type of dendritic cell, and we'll talk a lot about that today. But the important thing here is that antigen is not soluble and just held on the surface. It's taken up and converted, it's cut up by the proteasome or by proteases in the endocytic pathway, so that it's cut up into little peptides, and those are presented to T cells by professional antigen-presenting dendritic cells. Okay, so if we go back to all of these different colors of T cells basically mean they have a different T cell receptor, so they're specific for some antigen. And so when they get activated on a professional antigen-presenting cell, here are our antigen MHC complexes on the surface of this dendritic cell. They undergo proliferation and differentiation to form effector T cells. That's sort of like what's happening in a germinal center where you get proliferation, but there's some differences here. T cells don't change their T cell receptor. There's no somatic hypermutation in T cells. They differentiate into effector cells, they go and do their job, and the next lectures we'll talk about what those different jobs are. But then eventually they're all going to die off, or most of them are going to die off. And so you end up with a population of memory T cells. And again, these they haven't changed their T cell receptor, so they have the same TCR as this initial naive T cell. So when we talk about frequencies, you can say that 1 in a million of naive cells is specific for some antigen. When you're looking at the height of the T cell response, which we will cover again during the dynamics of the immune response, you're looking at a frequency that's 1 in 10, or 10% of all of your cells are effector T cells for that one epitope. And so this is about this is up to 100,000-fold amplification of that one clone. Now, after the effector response goes out and does its job, hopefully killing off the infection, then most of those cells are going to die. And then you get frequencies of 1 in 100. Now, this may not seem like a big difference, but this is 90% loss of T cells, or of these effector populations. The reason that this is important is because effector T cells are dangerous. We often say in this class that the most dangerous cells in your body are CD4 T cells, in particular, the helper cells, right? They control everything. If you keep those cells around long enough, they'll eventually do a lot of damage and end up killing you. And so you can't keep around large populations of effector cells. Okay, so the T cell activation in the lymph node is by these professional antigen-presenting cells. And you get just a very few naive antigen-specific T cells are going to recognize antigen, but then they proliferate so much that they form a very potent population. Now, what we've discovered is that after the pathogen is cleared, or even if it isn't, most of these effectors die off. And that comes back to the reason that these cells are too dangerous. So no matter what happens, this response is pre-programmed to die off so that the immune system is not going to kill the host. But when they do that, you leave behind this stable memory population. For CD8 T cells, it looks like it's absolutely stable for the rest of your lifetime. For CD4 cells, it may drop off a little bit, but essentially you have a relatively stable memory population so that the next time you get infected, these cells don't have to go through all the checkpoints. They've already made a response, and so it's fine. They can just go ahead and respond. So the first question we have is, how does the antigen get to the lymph node? How is it traveling there? Okay, so let's say you have an infection in your big toe. In this case, it looks like more the heel. That antigen has to get to the lymph node. Okay, in this case, it would be your popliteal lymph node behind your knee. And then that is the antigen trafficking part. How does the antigen get there? So typically, our peripheral infections occur when there's a breach in those physical barriers, either skin or mucosa tissue, there's some damage, and so now you can have an infection. So there's a chat question about why would prolonged effector activation kill the host? The answer is that in most of the cases, what's happening is that the T cells are producing cytokines which cause vascular leakage. And we can go into this in later in the semester when we talk about different applications. But essentially, during an infection, if you don't turn that response off, then you get intravascular leakage and loss of blood volume. Eventually, that leads to cardiac arrest, and you die. So in terms of infection, like in Ebola, it's called bleeding out. And it can happen if the infections are severe enough. Okay, so if we're getting back to our peripheral infection or introduction of antigen, the antigen is slightly different, right? It's for B cells, the antigen is delivered as C3b antigen complexes that are carried by macrophages expressing complement receptor 2. They take the antigen to the lymph node, and that's then passed off to B cells, non-antigen specific B cells, which transfer to follicular dendritic cells. For T cells, antigen is from the periphery is delivered as MHC antigen complexes on dendritic cells that get activated in these sites of infection. This is the primary way. Remember, anything that's smaller than a bacteria can diffuse through the lymphatics and get to the lymph node on its own. And so this happens for many viral infections. You just get the viruses trafficking there to the lymph node on its own. And then you have dendritic cells in the lymph node that will pick it up and perform the same job. But in either case, antigen is getting there differently. If it's not soluble antigen, it's getting there differently than for T cells versus B cells. Now, in this class, we're going to really talk about dendritic cells as sort of just a general dendritic cell, okay? But there are actually lots of different types of dendritic cells, and you don't need to know all of them. There are lymph node resident dendritic cells. So these include dendritic cells that don't express CD4 or CD8, ones that do express CD4, ones that do express CD8. I don't care that you know those differences. The main thing that I want you to focus on is there are dendritic cells in the lymph node that are called lymph node resident dendritic cells. And there are dendritic cells that come from the periphery, which are called migratory dendritic cells. Okay, so if the antigen is small, like a virus or proteins, it can get to the lymph node on its own, and then you're going to have these lymph node resident dendritic cells that can pick it up and present it to T cells. If the antigen is in the periphery, and these dendritic cells, which you have in all tissues, pick that up and get activated, they'll migrate to the lymph node and then present it to T cells. Now there's one other kind of dendritic cell, which is called the plasmacytoid dendritic cell. And this was discovered about 15 years ago. It's not great at antigen presentation. It can pick up stuff and it can recognize infection, but its main job is not to activate T cells. Its main job is to produce type 1 interferons. And you remember, type 1 interferons will poise cells to shut down should they become infected. But it's also one of the key cytokines for getting a T cell response. Okay, so there's one dendritic cell population I would like you to know. The other one is the CD103 positive dendritic cells. And that's because these are the main antigen cross-presenting cells. Okay, so these are the cells in your peripheral tissue that are picking up antigen in the endocytic pathway, feeding that into the cytoplasm by Sec61, where it's then degraded by the proteasome, fed back, and those peptides are then fed back through TAP into the MHC1 loading pathway. Now that's important because that's the main way that you're going to activate CD8 T cells, is by these CD103 positive dendritic cells. Those are the only two that I think you should really remember because they have implications for later when we're talking about how the response proceeds. Okay, so now let's say that you're a dendritic cell, either in the lymph node or sitting in the big toe, and you sense that there's something there. How do you take it up? Well, for class 2 presentation, right, MHC class 2 to CD4 cells, it's really going to be either sort of receptor-mediated phagocytosis, so you're expressing all of these proteins that we talked about in the very early lectures, the scavenger receptors, Fc receptors for antibody-bound stuff, complement receptor 1 for C3b coded things. That's going to cause dendritic cells and some phagocytic cells to take that up into the endocytic pathway. You're also having macropinocytosis. This is where the dendritic cells are just constantly sampling their environment. That's really anything, bacteria or smaller can be taken up by this pathway. These are going to be fed into the lysosome, or they're going to fuse with phagolysosomes, and eventually form the M2C, where you load these peptides, degraded peptides, onto MHC class 2. For CD8 T cells, you need to also do this, right? So when I was first taught immunology, we only really knew of viral infection, which was this virus comes in, it infects it infects a dendritic cell, and then that dendritic cell will see that, process those proteins from the virus, and then present those on MHC class 1. That actually doesn't make a lot of sense that that could be the only way, because then viruses would just evolve not to infect dendritic cells. We'd say, well, let's just skip those cells, and then they would never be recognized. But we now know that cross-presentation is very important for activation of CD8 T cells. And that's where these same pathways, receptor-mediated phagocytosis or macropinocytosis, are actually taking these antigens, they're degrading them, and then feeding the proteins into the cytoplasm, where they're then degraded by the immunoproteasome, and then eventually put back by TAP into the class 1 loading pathway. Okay, so this is how you're going to take up antigens, and this happens both for lymph node resident dendritic cells or dendritic cells activated in your big toe that are going to migrate to a lymph node. In particular, we sort of start to divide this by where these cells typically are. And the lymph node resident dendritic cells are better at activating CD4 T cells, where they're taking up antigens that make it to the lymph node. Mostly the cells that are going to activate CD8 T cells are going to be the peripheral lymph node or peripheral site migratory lymph node. So that doesn't. They're going to be peripheral migratory dendritic cells that then move to the lymph node. There are also other pathways that we don't discuss in this class. There are things like, instead of cross-presentation, there's something called cross-dressing, where dendritic cells can steal MHC from each other. I don't care that we cover this. This is trogocytosis. It's a different mechanism, and we're not going to cover it. But there are some other variations. So let's focus on a dendritic cell in the periphery that is going to take up antigen and then migrate to a lymph node. Okay, so we have our Langerhans cells, which are a dendritic cell that's in the skin. And there's a break to the epithelial layer where you have antigen coming in. Well, this could be a virus, it could be a bacteria, it could be even free protein. These dendritic cells will take up that antigen or pathogen, and they get activated. How do they get activated? Well, you have all of those innate receptors, right? You have toll-like receptors, Rig-like receptors, Nod-like receptors, and those are what the cell is using to sense that there's something here. And those Langerhans cells, when they do this, they'll stop taking up antigen and move to a lymph node. So here's our lymphatic duct leading through the efferent lymphatics, ultimately coming into the lymph node by the afferent lymphatic. And these dendritic cells will come in and move to the T cell zones of the lymph node, where they can activate T cells. So they can do this if there is a peripheral dendritic cell. They can take that up in the skin or wherever there is a site of infection. Or if the antigen travels on its own to a lymph node, it's going to go in through the lymphatics as a free soluble thing. It's going to travel through that follicular reticular network, which is a leaky pipe. And that leaky pipe is going to leak out the antigen, and the tissue resident dendritic cells will pick it up in the lymph node and essentially fulfill the same roles as the cells that migrated there. Now there's some debate here of what is the form of antigen. And we do know that resident dendritic cells in the lymph node can actually steal antigen from follicular dendritic cells. Like the follicular dendritic cells are holding antigen to activate B cells, but these other sneaky resident dendritic cells can actually go and steal it from the follicular dendritic cells, and they can get antigen that way. Okay, so how does the dendritic cell know? Okay, there's an infection here, I need to go to the lymph node. Well, the immature dendritic cells in your periphery are expressing all of these different infection sensors, right? These pathogen-associated molecular pattern receptors. So this can be scavenger receptors like DEC205. It can be toll-like receptors that are going to sense microbial PAMPs. For bacteria, remember, it's going to be lipids and sugars. And for viruses, it's going to be mostly nucleic acids. When those PAMP receptors are engaged, it signals that cell to change its chemokine receptor expression. And the chemokines are the proteins, the intercellular messengers that tell cells where to go in the body. Okay, and so a immature dendritic cell that's just sitting in the periphery is going to have chemokine receptors that say, yeah, stay here. Okay, so CCR1, 2, 5, 6. There's various ones depending on which tissue it's in. But once it gets these PAMP receptor signals, it then downregulates those signals that are telling it to stay in the tissue, and it upregulates a chemokine receptor called CCR7. This is a key chemokine receptor, and the reason that I bring it up is because this is the same chemokine receptor that tells the T cells where to go in a lymph node. So this is telling the T cell, or sorry, it's telling the dendritic cell, you're going to go and look for T cells. Okay, when it does that, it enters the lymphatics and goes to the lymph node. Okay, so the immature dendritic cells, before they get activated, express all of these PAMP receptors, and they're constantly sampling the environment by macropinocytosis. Once it recognizes the PAMPs, that results in the dendritic cell becoming mature. Okay, and it does a couple of things. First, stops pinocytosis. It says, okay, I've got something, I'm going to go with this. And it makes a lot of sense, because if it kept sampling the environment, you'd start taking up self-antigens, and that could be very bad. It also starts expressing the other components of the immunoproteasome, right? So it changes the cap and some of the enzymatic subunits. That just makes it good at processing antigens for putting it into the class 1 pathway. And then it finally upregulates this chemokine receptor, CCR7, which tells it to go to a lymph node. And in the lymph node, the follicular reticular cells are expressing CCL21, which is the ligand for CCR7. So it's basically saying you're going to go to lymph node and then go along the follicular reticular network. Now, in the process of migrating to the lymph node, right, which is controlled by this protein CCR7, it's going to start doing a couple of other things. It enters through the afferent lymphatics, that's into the draining lymph node, and these PAMP receptors and CCR7 signals are going to tell that dendritic cell, you're going to start expressing costimulatory proteins and adhesion proteins. The adhesion proteins just enable this cell to stick to other cells better. Okay, so this would be like things that recognize the intracellular adhesion molecules, ICAM1 and 2. And it's going to also express costimulatory proteins, so that if a T cell comes and recognizes antigen on that cell, this dendritic cell is essentially saying, yes, I got a PAMP signal and I have an antigen, so that gives you that gives the T cell a positive signal that that cell can go and make a response. Now, ultimately, what happens is you have this mature dendritic cell in the lymph node. It's been stimulated to express, the sticky proteins, the adhesion proteins, and co-stimulatory proteins, and eventually that rare 1 in a million CD4 or CD8 T cell will come along and recognize antigen on that dendritic cell and becomes activated. Now, for it to be activated, it absolutely requires this co-stimulatory protein. Otherwise, it undergoes an abortive process. And that's usually called anergy. Now, in reality, T cell anergy, which is the opposite of energy, my accent doesn't make it easy to distinguish, but anergy is the absence of energy, meaning it can't respond. Anergy really has only been described for CD4 T cells. CD8 T cells have a different mechanism for turning off the response. But what happens is here's our antigen presenting cell. And it's taken up antigen, it's processed it, it's put it onto MHC proteins. In this case, we're not distinguishing between which ones. It's got a PAMP receptor signal, so it's upregulation co-stimulatory proteins and adhesion proteins so these cells can stick together. And the T cell will come along and get both the antigen signal, okay, which we typically call signal 1, and the co-stimulatory signal, which we call signal 2. This is very similar to a B cell, right? For a B cell, it would get the antigen signal by binding to the antigen part of the complexes on the FDC. But it also got this other signal from the CR2, which is in B cells we call the co-receptor. In T cells, it's a different series of proteins, but we call those the co-stimulatory proteins. So that's signal 2. If you get both of those signals, plus some cytokines, you get activated T cells. But what Bill Allen and Brian Evervold and others have shown is that if you have a immature antigen presenting cell, so a cell that's taken up antigen, but it hasn't got a PAMP receptor signal, it doesn't upregulate co-stimulatory proteins, and so the T cell, even though it recognizes the antigen, comes along and says, well, there's no co-stimulatory signal, so this is probably a self-antigen, I should not make a response. And so for CD4 T cells, that just means they become anergic. Eventually those cells will die off, they'll become apoptotic, and they get eliminated. So this is one way that you maintain peripheral tolerance, is that if your tissue-resident dendritic cell takes up a self-antigen and presents it, but it hasn't got a PAMP receptor signal, then any self-reactive T cells that recognize that self-peptide are going to get deleted. Okay, so then often in textbooks it gets a little confusing because you think about, it'll often say antigen-presenting cells include dendritic cells, macrophages, B cells. It can also include T cells and some other groups. But really only dendritic cells can start a T cell response, okay? And there's some reasons for this. First of all, dendritic cells are constantly taking up antigens by macropinocytosis. If they get PAMP receptor signals, they automatically turn on high MHC expression, high co-stimulatory protein expression. And so that makes them really good antigen presenting cells, okay? So once this cell gets activated, it's always, first of all, it's always taking up stuff. Then it expresses high levels of MHC, and then it further upregulates co-stimulatory proteins. Now, the reason that we include macrophages and B cells as antigen-presenting cells is, first of all, macrophages can take up, obviously, their job is to phagocytose things, hence the name. And so they're taking up stuff from the environment. But they don't really express MHC very highly, but they can. If they get, bacterial, lots of PAMP receptor signals, they get some cytokine signals, they can start to express MHC. Well, where did the cytokines come from? The cytokines came mostly from T cells, and so you have to have a T cell response for the macrophages to be able to start presenting antigen. They also will start to express co-stimulatory proteins, but they have to already have gotten the signals. So you're still talking about, you had to generate a T cell response to signal macrophages. Yes, you should start doing this. So once the response is going, macrophages can be antigen presenting cells. B cells can also do this. Okay, they're actually pretty good at stimulating T cells. They do express MHC. If they get signals, particularly cytokine signals from T cells, they can upregulate co-stimulatory proteins. But the problem here is that B cells are taking up antigen on their B cell receptors. And so you got 1 in a million B cells is actually going to recognize antigen. And so although they can take it up very well, the ones that are actually antigen specific are very rare. And so, yeah, it can, but it's not something you would rely on, because you'd have 1 in a million B cells trying to take up antigen and present it to 1 in a million T cells. It gives you 1 in a million chance of actually stimulating something. So we include them because we know they can do it, but they're sort of waiting for the response to happen. Now, having said that, once the B cells got activated and they start proliferating, sure, then you have lots of B cells that are antigen specific, and they can certainly be antigen presenting cells. So really, when you're talking about the types of antigen presenting cells that can start a response, it's dendritic cells. Once the response starts kicking in, you have either T cell proliferation and production of cytokines, macrophages can do this, or as the B cell response kicks in and you start getting lots of antigen specific B cells, they can kick in. It's really dendritic cells that are the ones that start the response. Okay, so any question about how the antigen's getting to the lymph node and how it's getting onto cells that are going to present to T cells? So the question is, would cancer cells or virus-infected cells suppress expression of co-stimulatory proteins to induce T cell activate or to induce T cell energy? Absolutely. The cancer environment is very immune suppressive, and it inhibits dendritic cells from it can inhibit them from presenting antigen. It can suppress the expression of co-stimulatory proteins. When we talk about cancer in the last part of the semester, we'll cover all the way that cancer cells screw up the response by doing all of these different things. Okay, let's move on to how the T cells get to the lymph node, right? The antigen's coming in either as free antigen through the follicular reticular network, where it's picked up by the resident dendritic cells, or dendritic cells activated in the periphery are going to migrate through the lymphatics to the lymph node, and they can activate T cells. But the T cells are coming in a different route, right? The T cells are not coming from the big toe, they're coming from the circulatory system. So the T cells are going from the blood into the lymph node, and then eventually back through the thoracic duct, where they're cycling between the blood and lymphoid tissues. But B cells and T cells both enter lymph nodes the same way. That's through the high endothelial venules. There are the follicular reticular cells, which actually surround the high endothelial venules, are making the same cytokine that we just talked about, CCL21. That's the chemokine that's going to bind to CCR7. So the HEV, the high endothelial venules, are actually in T cell areas in the paracortex. So T cells that come into a lymph node, they're right in the right place to start looking for dendritic cells with the right antigen or epitope on their surface. Remember, B cells had to go to the cortex, they had to go to a B cell area. And so they have additional signals and additional chemokine receptors that tell them to go to the B cell areas. And we don't cover those in this class. So if we look at a lymph node, this is a cross-section of a lymph node. Here's our afferent lymphatics, where antigen is coming in, or dendritic cells. Here's our efferent lymphatics, where cells that didn't get activated are going to leave. And then we have our B cell zones in the cortex. That's in yellow here. And you can see this is a germinal center, so obviously there's a response going on. Eventually, as the response is cleared, that germinal center goes away. But here's our T cell zones, right, in blue. This is where the T cells are going to get activated. So the T cells enter the lymph node by the high endothelial venules, okay? So if this is our cells, this would be the T cell. It doesn't go to the B cell zone. The B cells get additional signals to go up there. So they come in and they start looking for antigen. Now remember, the high endothelial venules are post-capillary structures. And so here's our blood supply to the lymph node, right? There's our artery coming in, there's our vein going out. So even in the lymph node, we have a capillary bed. Now post-capillary bed, you have these little, they're drawn here as little bulbs on the veins, right? Those are the high endothelial venules, and they actually are highly invaginated, and they express some unique ligands that T cells and B cells use to get into the lymph node. And when you stain these, and here's a staining of a lymph node, and this marker on the HEV is shown in red. You can see you have lots of these different high endothelial venules, and they're sort of spread out through the paracortex region. And their job is to allow B cells and T cells to enter the lymph node. Now, a couple of things. They're post-capillary, so they're lower pressure, right? If they're pre-capillary, they'd be on the arterial side, and that's the high pressure side. So you want it on the low pressure side. That allows the cells to slow down. They're highly invaginated. That provides lots of surface area for the T cells and B cells to stick to. And they express some unique glycosylated proteins that only exist in high endothelial venules that allow the T cells to stick even better to those sites. So we have to remember how our cells, what are the signals for cells to enter the lymph node via the high endothelial venules. And so we typically draw this as a four-step process, right? There's a rolling step, and this is mediated by sort of low affinity interactions. Now that in the high endothelial venules, the cells there express a unique glycosylation pattern called Sialyl-Lewis X. And this is recognized by a protein called L-selectin. L-selectin recognizes this glycosylation pattern on the high endothelial venules, allowing it to stick, but it's a relatively low activation or low affinity interaction. What happens is that these naive cells express CCR7, right? Which we've seen before. That's the chemokine receptor that peripheral dendritic cells will start to express once they get activated to tell them to go to the T cell zones. Well, it makes sense that you'd use the same receptor to tell the T cells where to go. So the follicular reticular cells around the high endothelial venules are going to express the ligand for this. And this signaling through CCR7, which is a G protein coupled receptor, will tell this cell, okay, you're going to change a protein called an integrin, in this case, LFA1, from a low affinity to a high affinity form. Okay, so this basically makes it stick wherever it's at. And these cells will just continue to follow those chemokines and go between the high endothelial venule endothelial cells, and they will migrate towards whatever cells are producing CCL21. I don't think you need to know CXCL12. Okay, so this is telling the T cell, follow this trail. Every time it's actually binding to CCL21, it tells the T cell that's the way you want to go. So it's a sort of a ratcheting motion. We often call it ratcheting because they're going to do this, bind, and they get a more signal, and they're going to bind higher in that direction, sort of like crawling. Now, in these high endothelial venules, there is this glycosylation pattern called Sialyl-Lewis X, okay, which is on a number of different proteins. The glycosylation is on a number of different proteins. So here's our high endothelial venule. It can express Sialyl-Lewis X on a couple of different proteins, including CD34 and glycam-1. These are just general adhesion proteins. And so L-selectin which otherwise is sometimes referred to as CD62L, on the naive T cell is going to recognize this carbohydrate structure. Now the T cells, once they get the CCR7 signal, are going to change their LFA1 from a low affinity form to a high affinity form. And this is a structural movement, right? A conformational change in the protein, which allows LFA1 to bind very strongly to an adhesion molecule called ICAM1, intracellular adhesion molecule 1. And this is, and particularly when we're talking about the high endothelial venules, this is the main protein that's being recognized by the integrins to tell that T cell bind strongly here and then move in that direction. Now, as I said, the chemokine receptor is a G protein coupled receptor, and so it's G protein coupled receptors typically will go turn this on and then they get deactivated. And so this integrin is cycling between low affinity and high affinity forms every time it gets a chemokine receptor signal telling it where to go, okay? Hold on just a second. Okay, so the key proteins here that I would like you to know are L-selectin, which is going to recognize Siala-Lewis X. That's the low affinity rolling and sticking interaction. There's a chemokine receptor that signal that says, okay, we're going to have a strong binding affinity interaction. And so that's this LFA1 binding to ICAM1. So when we include those in this sort of rolling and sticking and then activation, tight binding, and diapedesis, you have, here's our T cell that's rolling along, and it's using L-selectin to bind to the Siala-Lewis X structure. That's the low affinity interaction that causes this rolling. The next step is that the chemokines that are being produced inside the lymph node are actually being transported and held on the surface of other cells by fibrin-like proteins, okay? And so in this case, CCL21 is being held on the top of these cells, and as this cell is rolling by, it's going to recognize that CCL21 by CCR7, which is the chemokine receptor, and that's going to activate LFA1. LFA1 then is going to bind very strongly to the ICAM1, allowing that cell to say, stop, this is where I'm getting off the freeway. And then because there's lots of CCL21, it's essentially going to follow that path to the highest concentration of CCL21. Okay, now there's a lot more involved in this. There's reorganization of the mitochondrial of the mitotic organizing center. There's lots of actin polymerization and sort of moving the cell, but it's directional. Okay, and I don't really care that we go all the way back to cell biology and sort of say, how do cells move? It's more about them just following the trail of the chemokine. Okay, so here's our lymph node. The T cells come in through the HEV, right, which is this post-capillary structure. It's one of the things I don't like about Janeway's textbook is he just draws them as circles, but they're not really circles. They're very organized structures. And that's in response to CCL21. This requires those other proteins, L-selectin, or CD62L, and LFA1. The migratory dendritic cells are coming in through the lymphatics. The T cells are coming through the HEV, but they're both expressing CCR7, so they're both following the trail of CCL21, and eventually that's going to cause them to crash into each other, okay? So migratory dendritic cells are going to come in looking for CCL21. The resident dendritic cells are already there. They're already following along the follicular reticular network, remember, on the outsides of these tubes. And they'll pick up antigen in the lymph node, but eventually T cell is going to run into them too. because they're expressing the same, same homing receptors. Okay, so now the T cells came into the lymph node by the HEV. And they're looking for antigen. And here these green cells are looking for antigen. They don't see it, and they leave. And so this actually should be medullary sinus. They're going through the medullary region, out through the medullary sinus. And they're just leaving like they normally would. They go to the next lymph node and see if they see antigen there. Okay. So after the naive T cells come to the lymph node, they start to lose expression of CCR7. It's like a clock, really. And the way that this is easiest to explain is that there's two competing chemokine receptors. CCR7, which is telling the cell to go to a lymph node, and a S1P1 receptor, which is a different chemokine, which is telling the cell to leave the lymph node. Okay, so when a cell is in the blood, it's expressing CCR7 because it wants to go to a lymph node. Once it gets to a lymph node, it loses CCR7 and starts expressing the S1P1 chemokine receptor, so it leaves and it goes to the blood. Okay, so this is how the T cells are constantly cycling in and out of lymph nodes. If no antigen recognition occurs, naive T cells just migrate along the S1P gradient to the medullary sinus, and then they exit. Okay, so they're coming out through the efferent lymphatics. However, see these blue cells over here, if they actually do recognize antigen, then they do not lose CCR7, and they don't decrease S1, they don't lose CCR7, and they don't upregulate S1P1 receptor. Sorry, the way I've written it says they decrease S1P1 receptor, but they actually just never make it. So they don't leave the lymph node. Okay, so just remember that CCR7 says go to a lymph node. The S1P1 receptor says leave the lymph node. So if you don't recognize antigen, you express the S1P1 receptor, you leave. If you do recognize antigen, you want to stay there, so you keep CCR7. Is that clear?
Dr. Blackman?
Yes.
So the chemokines direct the cells to the area of high concentration of that chemokine.
Correct.
So are these chemokines binding to these receptors, and then they just know where the high concentration gradient is?
Well, this is, as I said, this is sort of ratcheting movement where you make a recognition event, the cell moves that way. Then it's going to, if that's the right high concentration direction, then most of the signals are going to tell the cell, yes, keep coming this way. So it's turning on the high affinity integrin, and then as that turns off, it's moving towards whatever direction it's getting the chemokine from. And we're not going to go over leading edge and trailing edge cell movement, because I think what's more important to focus on the immunology parts of this.
So this is the same mechanism from when they're in the periphery all the way to this high endothelial venule, and then as well as when they migrate from the paracortical to the medullary sinus?
Right, it's just a different chemokine receptor. Remember, when they're in the blood, they're just sort of floating along. They're not really using this mechanism. But to get from the blood, where they're floating, into the lymph node, they have to use a chemokine signal to go where they need to go. Now, let's say a T cell gets activated, and it proliferates, differentiates into effector cell, and then it's going to leave the lymph node to go and kill infected cells. Well, it's going to use different chemokine receptors to tell it where to go in that case. Okay, so things like IL-8 and other chemokines that are going to direct the cells, yeah, here's the infection, come look for infected cells here. Well, I'm trying not to go too heavy into the chemokine receptors. I'm just focusing on the main two, the one that tells you to go into a lymph node and move along the follicular reticular network, and the one that tells you to leave the lymph node and go to another one to look for antigen. Okay, so now you got antigen recognition, right? These T cells recognize antigen on a dendritic cell, And they got co-stimulatory signals, they got some cytokine signals, and they're proliferating. And as they're doing that, they're also changing their transcription pattern to differentiate them into effector cells so they can actually do their job. Now there's some differences here, right? For B cells, they would initially get activated on follicular dendritic cells, and then there's some steps before they would start to proliferate. For T cells, they don't have to wait for B cells. They just start proliferating. They got the signals from dendritic cells, so that's the checks and balances here. T cells, though, do not do two things that B cells do. There's no somatic hypermutation, and there's no isotype switching, right? T cells don't adapt based on their receptor. T cells adapt by changing the cell, okay, rather than the receptor. Now, once this happens, your effector cells leave to go and do their job. Now, there's a couple of things that are wrong with this actual figure from Janeway. He has these cells moving out through the medullary sinus. Well, this is an older version. Think about that for a second. You've spent all of this effort to generate a very rapid and efficient T cell response, so you need to deploy that as rapidly as possible. So the cells don't actually leave there, because then it would have to percolate through lymph node, through lymph node, through lymph node. You know, in humans, you have hundreds of lymph nodes, eventually getting to the thoracic duct and the blood. That would be very inefficient. Instead, what they do is they leave here, okay? So the effector T cells leave via the efferent or through the high endothelial venules, because the lymphatics are too slow. The second thing that's wrong with this figure is that it's basically saying all the cells leave the lymph node, which is not true. Remember, we had one cell type that was important, one T cell type that was important in the B cell germinal center. Anybody recall what that is? Anybody respond? You have to raise your hand. What was that one T cell that was important in the B cell area?
The T helper one?
Okay, there's T helper cells. So first of all, our B cell and our T cell, so we'll go get here, right? That's the initial signal for B cells to get a CD40 signal from a T cell. So that's the key thing. The other one is that some of these T cells, remember we have these T follicular helper cells that are in the germinal center that are providing those B cells with the additional CD40 signals to go back to the dark zone. So if we're just talking about CD8 T cells, yeah, great. Everybody leaves. They're actually leaving through the high endothelial venules. But when we talk about CD4 cells, some of them are going this way too. Okay, and we'll get into that in the next lecture where we talk about what CD8 T cells are going to do and what CD4 T cells are going to do. Okay, so now we have to start talking about what are the signals for a T cell to get activated. And to understand this, we have to understand what are the things that are binding those cells together, okay? So the first step is you have to have the right adhesion molecules for T cells to bind to dendritic cells, okay? And so these are those tight binders, right? LFA1 and ICAM1. They're being controlled by CCL21 signaling through CCR7. So this is telling the cell, okay, you're going to stick tightly to that dendritic cell. Now, in reality, LFA1 and ICAM1 are actually on both T cells and B cells, but we typically only draw the LFA1 on the T cell side and the ligand for it, ICAM1 or ICAM2 on the dendritic cell side. But in reality, they're on both. And that's because if you remember, what chemokine receptor are the dendritic cells expressing? Well, that's CCR7, so that's telling them to go look for a T cell. Well, there's a mutual expression of both of these. Now the T cell and the dendritic cell are both going along the outside of the follicular reticular tubes, right? These leaky tubes that allow antigen to get into the lymph node. So that increases the probability that they're going to run into each other. They're also migrating to the same chemokine gradients, That further increases their chances of getting together. And they're also expressing the same adhesion proteins, okay? So these allow the cells to, it increases their probability of colliding together, and then they express the same set of adhesion molecules so they can stick together. Now, some dendritic cells have different adhesion proteins. We're not going to get into that. We're just going to treat dendritic cells essentially all the same. The reason that some have different adhesion proteins is some are better at activating CD4 cells versus CD8 cells, but I don't think that that's terribly important for just understanding initiation of the immune response. Okay, now we had talked about this sort of CCR7 signaling activating LFA1. But remember that if a T cell recognizes antigen, you also want it to stop. And so what happens is if here's our T cell on the top, and our antigen presenting dendritic cell on the bottom. If the T cell receptor recognizes antigen, that also activates LFA1 to the high affinity version. And that tells the T cell, here, we found something, stop right here. And now we're going to see if we can get full T cell activation, okay? And so there's sort of this just browsing thing where the T cells are bumping into dendritic cells, looking for antigen. There's a aha moment, okay, there's some antigen there, and then it sticks. And that sticking is important because now to get full T cell activation, it has to rearrange all of its proteins. Okay, so when we're thinking about T cell activation, we've covered two signals, but there's a third signal that's important here. And this has to do with you want the cell to be antigen specific, and that's accomplished by signal 1, which is the T cell receptor recognizing the antigen on MHC1 or MHC2 proteins. The second thing is this dendritic cell had to have gotten signals to say, yes, express costimulatory proteins. And the main ones are the B7-1 and B7-2 proteins. Those are going to bind to the costimulatory protein CD28 on the T cell. Okay, so that's signal 2. That tells the T cell, yes, this antigen presenting cell has seen some PAMP signals through its PAMP receptors. So it upregulated costimulatory proteins. This is very likely an infection. But now you have to sort of tell that cell what to do, right? Remember, we have those T cells are going to be responsible for telling B cells which isotype to make. They're going to make the cytokines. They're going to direct the response or activate accessory cells. So you have to sort of tell it what kind of response you want this to be. And so the dendritic cells got the PAMP receptors engaged. Remember, that results in them making certain cytokines. If it's a bacterial PAMP, it's going to make more IL-12 and cytokines that are going to direct an extracellular response. If it is a nucleic acid that's being recognized by Rig-like receptors or some of the Toll-like receptors, then it's going to give a signal that says this is an intracellular thing and tell the T cell, what kind of T cell do we want to be, okay? Now, one thing that we know, so this is the third signal, which is a cytokine. Signal 1 is antigen specific. Signal 2 is, yes, it is an infection because I got a costimulatory protein. Signal 3 is what kind of infection is it? Okay, so it's really modifying it. But there's this other case that happens, and it's worth noting because if you have strong enough Toll-like receptor signals to the T cell, it just kind of says, go ahead, make a response anyways. Well, when would that happen? When would you get strong Toll-like receptor signals in the lymph node? Well, it would be during a systemic infection, right? So if you've got a systemic infection, you've got virus or bacteria in the lymph node, basically saying, throw everything at this, including the kitchen sink. You don't need to worry about getting cytokines. You get a Toll-like receptor signal, just keep going, okay? So those are sort of the three things. And we often call these, we often separate these into something called activation, survival, and differentiation. But activation is just Yeah, I'm antigen specific. I recognize my antigen. Survival really means don't become allergic and don't die. Okay, so that the co-stimulatory signal is basically saying, yes, this is in fact an infection, so we're not going to kill you off. And the differentiation is really what kind of T cell is going to be most useful. Do we want a Th1 response for a viral infection? Do we want a Th2 response for a parasitic infection? That's going to tell us, excuse me, what kind of response to be. Now, there are some exceptions to this, and we covered one, strong TLR signal. That means a systemic infection, which just says go ahead, okay? If you get a strong enough signal 1 and 2, you can overcome the requirements for a signal 3. Well, when does that happen? Well, it does happen with some superantigens. And these are proteins expressed by bacteria that force engagement of T cell receptors with MHC. And that can activate a T cell, but that's not terribly common. Some of the staphylococcal varieties will do this. Or you can do it with anti-CD3 antibody, but that's not something you do in vivo. That's something you do in a tissue culture dish. And you can get activation of T cells in those ways, but that's not really, most of the time, it's not physiologic. Okay, so now what we have to focus on is the different proteins that are being, or that are sticking these cells together. And the first thing that you should notice is that they're different sizes. They stick way out. Some of these larger proteins are they're very large and they stick out very far from the cell surface. They're highly glycosylated and they're low affinity binding interactions. Now, what's typical for these very large proteins is that they have phosphatases attached to the intracellular parts of them. And so that's going to be important because what you want to do in any kinase activation signaling pathway is put those on the outside so they can't inhibit the kinases and get all the kinases together. Now, sort of at intermediate level, we have the sort of adhesion molecules that are facilitating tight binding, LFA1 and ICAM1, right? So this is, as these are tight binders are getting together, they're actually forcing these big proteins out of the way, okay? Now the actual MHC or TCR interactions are very small proteins, relatively speaking. They don't stick out far from the surface. So to actually get TCR that are going to recognize MHC, you've got to force everything else out of the way so that these smaller proteins can get together and actually recognize each other. Now what happens is that you get recruitment and activation of kinases. I'm just going to put a K on here. The kinases are opposed by the phosphatases. So as you push things out, the phosphatases get excluded and you get just the kinases. And that's what allows for T cell signaling to happen. When I was first taught immunology, it wasn't clear that how this worked. It was thought that there was some conformational change in T cell receptors. We now know that's not the case. It's just pushing the phosphatases out and recruiting the kinases in. Okay, so this is sort of what that looks like. As you get this activation, these big proteins with the phosphatases get stuck way outside. And then you have this ring of adhesion molecules that allows just the very small, actually, antigen recognition things like TCR, the co-receptor CD4, the co-stimulatory protein CD28 to recognize their ligands on the antigen presenting cell. Okay, so this is sort of a cross-sectional view. But this was done in Mark Davis's group at Stanford quite a while ago, is that we started to realize there's a dynamic change, okay? So the dendritic cells are going to recognize the antigen presenting cell, or they're going to bind to it, and then they're sticking to each other. And so what initially happens is that the LFA1 ICAM1 interactions are what is sticking the T cell to the dendritic cell. Now, what these pictures are showing is the interface between a T cell and a B cell. So imagine two balloons pushed together. It's the part where the two balloons are meeting. That's what you're looking at, okay? Now, as that happens, this T cell and dendritic cell, they're interacting, and the LFA1 components are being pushed to the outside, okay? And it focuses all of the peptide, MHC, the TCR, stimulatory proteins, all go towards the inside of it. So there's this reorganization of all of the TCR recognition components, as well as the antigen presenting components on the dendritic cell. Now it's doing this, there's a lot of other steps in this. These are preformed TCR in lipid rafts. I don't go into that a lot in this course, just because I think it's details that don't really help you understand the immune system. This is all tied to cytoskeletal rearrangement. So this is an active process inside the cell. So the whole point of this is the initial interactions between the T cell and the dendritic cell are the adhesion molecules, LFA1 and ICAM1. But eventually, you're pushing the TCR together, or the MHC antigens, to become the central part of it, okay? Bring them all together. Okay, so we don't, at this point, we haven't distinguished much between CD4 and CD8 T cells. There's some difference in the co-receptor, right? That the CD4 cells have this monomeric protein, CD4. The CD8 protein is actually a dimer, an alpha-beta dimer. For CD8, it binds the alpha-3 domain. For CD4, it binds the beta-2 domain. But their job is really just to stabilize the TCR, right? It increases the avidity of the TCR for MHC peptide and stabilizes that interaction. So if it is an antigen-specific recognition, you can get activation. The other thing that these co-receptors are doing is bringing in the very first protein receptor tyrosine kinase. In this case, it's a protein called LCK, okay? This is the first step in T cell activation, but in order to get this to happen, you have to have that brought in by the co-receptor. Now, we're gonna go through some of the signaling here, and I wanna point out where it's similar to B cell receptor signaling and where there's a little bit of difference, okay? So the first step, here our T cell is down here, and our dendritic cell is up here. The dendritic cell's got MHC class, in this case class 2, with a peptide. And here's our T cell receptor. It's going to come in and recognize that. Now this co-receptor here, CD4, it's not an arrow, that blue line is actually a receptor. That's the co-receptor. That's going to bind to MHC class 2 to stabilize that interaction and bring in this kinase called LCK. Now, if this was just a T cell floating around and that CD4 bumped into the TCR and brought in the kinase, it wouldn't get activated because you haven't excluded phosphatases. So even if it was able to phosphorylate something, phosphatases would still be there and deactivate it. But now because you push those big proteins to the outside, the phosphatases are not there, and so the kinases can do their job, okay? So the first step is that once this recognition occurs, LCK will start phosphorylating the ITAMs here in orange on the T cell associated proteins. Now the main ones are the TCR zeta, right? There are other ones, gamma, delta, and epsilon, but the TCR zeta ones, Helen Kirsch and others showed that there's 6 tyrosines that are phosphorylated on that. Those are the critical signals for T cell activation. So, okay, and additionally, and again, it's just the exclusion of those big proteins with the phosphatases that allows this to happen. Now what happens? Well, LCK phosphorylated ITAMs on CD3 zeta here. And that recruits a protein called ZAP70. It's just another kinase, okay? And that then phosphorylates other proteins. The main one is this LAT protein. And it's called LAT because it was described as the linker for activation in T cells. And it's actually just a scaffold protein. It doesn't really signal anything on its own. And it's actually tethered to the membrane. Here it's sort of drawn in the cytoplasm, but you can see here it's stuck to the membrane. That gets phosphorylated and brings in lots of other proteins. And this is where T cell activation really happens, okay? The main ones that we'll talk about are phospholipase C gamma and the kinase cascade, okay? Those are some of the main ones that eventually are going to result in changes in cellular signaling, okay? That ultimately lead to activation of transcription factors and T cells being differentiated into effector cells. Now these wiring diagrams get a little complicated. And so what I've done is broken this down into steps. And this is really what I'd like you to know, right? Is what are the steps in T cell activation? And we started off with the initial activation, LCK. So you remember in B cells, there was the initial activation, right, was by FIN or SYN, you know, the other proteins. Those can sort of compensate for LCK, but they're not as important. LCK is the main one. So that's going to bind, the CD4 is going to bring that in, or CD8. It's going to phosphorylate the CD3 zeta, which brings in ZAP70, okay? ZAP70 phosphorylates LAT, and then it has all these other proteins that bind to it, okay? Now, what we don't talk about a lot here is the co-stimulatory proteins. But this is essentially doing the same thing that the complement receptor 2 is doing in B cells, except we just call it co-stimulatory instead of co-receptor, mainly because we already have a co-receptor. So you're really following this pathway. 1, this is going to phosphorylate these, 2, 3, 4, and then we move on to the next set of things that are happening. So one of the proteins that's going to bind to LAT and become activated is phospholipase C gamma. Okay, this takes diacylglycerol, or sorry, it takes phosphoinositol diphosphate, and will cleave it into 2 proteins. Right, it's taking a lipid from the membrane, and it's cleaving into IP3 and diacylglycerol. Now that doesn't seem like a lot, right? Just cleaving a lipid, but those activate 2 different pathways. The first is IP3 stimulates the calcium flux into cells. Calcium flux from the endoplasmic reticulum stores. This also activates the influx of calcium from outside the cell through other pathways. The main thing I want you to know is IP3 is opening up calcium release. And that leads to activation of NFAT, or nuclear factor of activation in T cells. Okay, so that's the text that I've highlighted in red. That's really the essential of what I want you to know about the results of IP3. For diacylglycerol, it's going to activate a protein kinase C theta. This will in turn activate NF-kappa B. For those of you who remember your cell biology, NF-kappa B is normally held in an inactive state by being bound to the inhibitor of NF-kappa B. Protein kinase C theta will phosphorylate the inhibitor of NF-kappa B, causing it to release, and then NF-kappa B is active and can go to the nucleus. Okay, but all I really want you to know is what's shown in the green text there. Diacylglycerol that comes from phospholipase C gamma activates this kinase, and that then results in activation of NF-kappa B, nuclear factor kappa in B cells. Okay, so we've got the two main transcription factors, and the third one is the MAP kinase cascade. Right, so one of the other proteins we talked about that binds to LAT is a protein called VAV. VAV is just a kinase. You get phosphorylation of other kinases that phosphorylate other kinases, This is an amplification cascade, right? So one kinase activates 10, those 10 activate 100 activate thousands. So once this gets started, you get lots and lots of kinases activated. Eventually, those phosphorylate other transcription components, CFOS and CJUN, which together, when they are activated and bind, form the AP1 transcription factor. Okay, so what do I want you to know? I just want you to know what's in the blue text, that VAV and SOS together will activate the MAP kinase pathway. There's also this little check here. To get the MAP kinases, you also need diacylglycerol. Okay, so it's the check that says, okay, yes, we're actually getting the other pathways activated, MAP kinases, go ahead, you can do your job too. Okay, so when we're talking about TCR signaling, lots of the events are proximal to the TCR. There's sort of things that are getting started that only happen when you exclude the phosphatases. But then there's a very limited number of set of things that will happen downstream of that. And that's where we'll pick up next week when we're talking about what are the cytokine signals to T cells. What does this result in, okay? And with that, I'll take any questions.
Dr. Blattman?
Yeah.
So when T cells are in lymph nodes or in the periphery, are they constantly attached to other epithelial cells, and so they're always rolling around looking to be activated?
No. So when you're talking about a naive T cell, when it's in the blood, it is not sticking to the endothelial cells of the bloodstream, right? Or your blood vessels, because they're expressing L-selectin, which is very specific for Sialyl-Lewis X. Sialyl-Lewis X is only in the HEV. So most of the time when a T cell's in the blood, it's just being pushed along with the current, okay? It's not sticking to anything. When a T cell is migrating within a lymph node, this is in response to chemokines, and so it's moving directionally. Did that answer your question?
So like when it's moving, but it's adhered to, like it has a low affinity interaction with the other cells, so it's rolling around inside the lymph node?
Correct, but it's at that point it's getting chemokine receptor signals, and so it's not just sort of loosely rolling, it's sticking, tight binding, sticking by LFA1. And it's moving in the direction of the chemokine, but it's sort of like clawing onto this, you know, LFA1's clawing on here, it's clawing on and moving sort of in a slow crawling way.
Okay, thank you.
Any other questions?
Actually, I have another question.
Mm-hmm.
So when there's differentiation of T cells, so these naive T cells, they have the ability to be a CD4 or CD8? Because I thought they were only double positive inside of the thymus.
So a double positive cell has the potential in the thymus to become a CD4 cell or a CD8 cell. Once it commits to one of those, it can't change. Well, there's a few exceptions, but it doesn't change.
So what is the differentiation?
So that's when you change from a naive cell to an effector cell. So a CD8 cell is a naive cell until it gets activated, and then it becomes a cytotoxic T lymphocyte, where it is able to go out and kill stuff. That's differentiation. Okay, any other questions? Okay, if there's no other questions, we'll see you next week.